Well, hello, my fellow pre-meds, and welcome back to another video. In this video, we're going to turn our attention to biological membranes. We're going to cover everything you need to know related to plasma membranes, including structure, transport, signaling, electrical properties, and more. Now, the concepts covered in this video are extremely memorization heavy. There's a lot you need to memorize. So what I'm going to do is I'm going to equip you and empower you with a solid understanding of the underlying mechanisms behind these concepts so that hopefully memorizing them will be a breeze. And I also recommend that you use our Anki cards associated with this slide deck to aid with memorization. So let's get started over here with the fluid mosaic model. Essentially, the fluid mosaic model posits that the plasma membrane is far from this simple bilayer of phospholipids, but it has these diverse macromolecules within it, including proteins, carbohydrates, and lipids. So that's the mosaic part of it. It's kind of a diverse macromolecules. And then the fluid part is that the components are continuously moving and shifting within the plasma membrane. So you can see this phospholipid, for example, at this location shift to this location, and then maybe in the next moment the protein that's here will shift over here. So it's constantly moving and there's fluidity to it. Now, importantly, the plasma membrane is selectively permeable. This is a very important concept that it will decide what goes into the cell and what stays out. So for example, large polar molecules cannot freely cross the plasma membrane, so they need special carrier proteins and channel proteins, as we'll discuss in subsequent slides. And then small non-polar ones can move freely. And then other functions of the plasma membrane include signaling and cell recognition. Now let's shift our focus to the major category of the macromolecules in the plasma membrane, which is membrane lipids. So the phospholipid is the backbone of the plasma membrane, and it's amphipathic, meaning it has a polar and a non-polar side. And then importantly, you can also see other lipids such as cholesterols embedded in the plasma membrane. And then because the phospholipid is composed of fatty acids, these fatty acids can either be unsaturated or saturated, and they play a role in the fluidity of the membrane, which influences the level of permeability of the plasma membrane. So let's take a look at what the phospholipid molecule looks like. So essentially, if you watched our video on structure and on the structure and function of lipids, we cover this at length, but let's just briefly mention it over here. So the structure of the triacylglycerol is shown over here. You have the glycerol molecule and 3 fatty acids, hence why we call it triacylglycerol. Now, phospholipids are very similar structurally to triacylglycerols. The only difference is that you're replacing that third carbon here, the yellow carbon. Instead of it now having a fatty acid, it's going to be attached to a phosphate-containing head group. And so that's what contributes to the amphipathic nature. So the polar side is this head group, and the non-polar side are the fatty acid tails over here. Now, if you take a look at this one is bent, so there's a kink to it, and that's because it is unsaturated. And when you have unsaturated fatty acids, they essentially increase the fluidity of the plasma membrane. And then also you have these cholesterols embedded, and recall, because cholesterol has a polar side, that hydroxyl group, and a non-polar side, the other side of it, it can interact with the plasma membrane. And the way it works is that it can play both ways in terms of fluidity. Like if we need to increase fluidity, cholesterol can increase fluidity. If we need to decrease fluidity, cholesterol can do that as well. And then this is just showing the structure of the plasma membrane again. So I really don't want you to think that the plasma membrane is just like this simple bilayer. It's a lot more complex than that. Now let's move on and talk about the proteins and carbohydrates that are embedded or associated with the plasma membrane. So there are 2 major categories of proteins. These include integral proteins and peripheral proteins. Integral proteins embed themselves within the plasma membrane, whereas peripheral proteins only interact with the surface through electrostatic interactions. And then you have glycoproteins and glycolipids, not extremely high yield on the MCAT. However, they do show up in terms of immune regulation. So the ABO blood type system is simply carbohydrates being recognized by the immune system. Now, let's take a look at the different types of integral proteins that you can have. So if we look again at this structure, you can see that this protein, for example, is spanning the entirety of the length of the plasma membrane, so all the way through. That is going to be an integral protein, but it's specifically going to be categorized as a transmembrane protein. Now, embedded proteins are also integral proteins. However, they're not going to span the entire length of the plasma membrane. So you can see over here, this protein, this globular protein, is interacting with the core of the plasma membrane. However, it's not spanning all the way through. And then finally, you have surface proteins or peripheral proteins that you can see right here. So these are interacting with the plasma membrane, but they're not entering into it. They're interacting with the surface via electrostatic interactions. All right, now let's talk about the functions of these proteins. So we're going to start by talking about ion channels. Ion channels are essentially transmembrane proteins that allow for the flow of ions across the plasma membrane. Recall, because these ions are charged, they can't just move across the plasma membrane on their own. Now, importantly, these proteins carry out what we call facilitated diffusion or facilitated passive transport. More on this in subsequent slides. Essentially, it doesn't require any ATP. It's just there to assist the movement of polar ions in this case. So let's take a look at the different kinds. We have ungated channels. So these continuously allow for the flow of charged atoms. So for example, potassium down its electrochemical gradient. And again, more on this in subsequent slides, but the idea is that these are ungated. They don't close. They allow for continuous flow. Voltage gated channels open or close in response to changes in membrane potential. So as you'll see when we discuss the nervous system in our biology playlist, the action potential is an example of the voltage gated channels. So essentially, once the plasma membrane potential reaches around like -55, that threshold allows for the opening of a sodium channel, which allows for rushing of sodium ions into the cell to allow for the generation of the action potential, and that is an example of a voltage-gated channel. Finally, we have ligand-gated channels, and these open or close in response to a signaling molecule or a ligand, and we'll see an example in the next slide over here. So as you can see right here, these are ligand-gated channels. This is an example of the neuromuscular junction. So you're going to have acetylcholine being released from that neuron terminal, and more on this when we talk about the nervous system in our biology slide deck. So you release acetylcholine, it's going to bind its receptor, and then it's going to open a channel. So that's an example of a ligand-gated channel. And this is an example of the GABA receptor, where you bind GABA and then you open a chloride channel, allowing chloride to enter the cell. So that's just another example of a ligand-gated channel. This graph over here implicates the ungated and voltage-gated channels. So you can see that the plasma membrane or the membrane potential is at -70, and that's established by potassium leak channels to a large extent, and we'll talk more about that in subsequent slides. Just know that these channels are continuously open and they establish that initial membrane potential. And then when you reach that threshold, you have the voltage-gated channels opening, and then you have a rush of sodium into the cell, and you have the action potential being generated. All right, now let's shift our focus from ion channels to receptors. We're going to first talk about enzyme-linked receptors. Enzyme-linked receptors, essentially, as their name implies, they contain a receptor on the surface of the plasma membrane, so on the outside of the cell, and then they have a transmembrane protein, and on the other side, they have an enzyme that's going to be involved in signaling. A common example that shows up on the MCAT are RTKs, or receptor tyrosine kinases, and we're going to look at them right here. So you can see that this is the receptor, and there are essentially two nearby receptors. Each of these will be considered a monomer. When you get the ligand, whatever it may be, it can be like a peptide hormone, when it binds to this receptor, essentially the two monomers will dimerize, so they'll join one another. And then what will happen is you will have autophosphorylation, so there will be a kinase over here. So that's where the enzyme comes into play on this end of the plasma membrane, so inside. And this kinase will autophosphorylate tyrosine residues over here. Recall from our slide deck on amino acids that tyrosine contains the hydroxyl group in its side chain, so it can be phosphorylated. And essentially that's going to trigger a signaling cascade downstream, so maybe increase CAMP levels or any other signaling molecule that can carry out a function within the cell. Now, one thing I didn't mention is that notice that the transmembrane protein over here, or the component of the protein that's spanning the membrane, is in the form of an alpha helix. So that's an example of a secondary structure. Recall from our structural hierarchy video of proteins. So whenever you see a transmembrane proteins, frequently it's going to be in the form of an alpha helix, and the amino acids composing that protein are going to be hydrophobic amino acids because they need to interact with the hydrophobic core of the plasma membrane. Now let's shift our attention to G protein coupled receptors. So they share some similarities with receptor tyrosine kinases or enzyme linked receptors. However, they are quite distinct and they're pretty high yield on the MCAT. So essentially they also contain like a receptor on the surface, and when a ligand binds to that receptor, such as a peptide hormone, for example, it will cause conformational changes to the G protein, which is a trimeric protein composed of 3 subunits within the cell, and that will carry out further signaling in the cell. So we can take a look at this diagram over here. You can see that this is the trimeric protein composed of a alpha, beta, and gamma subunit. And then notice the alpha subunit has something bound to it over here, which is GDP. Now what happens is when the ligand binds to the surface over here, there will be a change. So instead of you having GDP bound to the alpha subunit, now GTP will bind. So you can see that over here. Once that happens, the alpha subunit will lose affinity to the beta gamma complex over there, and then it can carry out further signaling within the cell. So notice that when it's bound to GTP, it's on its own and it can carry its function. After it's done, it will hydrolyze GTP into GDP, and once again, gain affinity to the beta and gamma complex and not carry out any further signaling. Now, what kind of signaling can it carry out? Well, it depends on what specific G protein you have. So if it's like a GS, then it's a stimulatory protein. It can stimulate adenylyl cyclase to increase the production of CAMP and carry out further signaling. And then if it's an inhibitory, if it's GI, then it's the opposite of that. So it decreases the production of CAMP. And then finally, we have the GQ type, which activates phospholipases, breaks down phospholipids into IP3 and DAG, which increases intracellular calcium levels and carries out further signaling within the cell. All right, now let's shift our attention to cell-cell junctions. So cell-to-cell junctions, the purpose of them is to maintain the integrity of the tissue, but also to allow for communication between neighboring cells. So there are 3 broad types. The tight junctions form a seal between adjacent cells, and they don't allow for water or solutes to leak through. So think about areas of the body where you really need tight binding between the cells. So think about the blood-brain barrier, the renal tubular cells in the kidneys. The next broad category, maybe not as high yield on the MCAT, are desmosomes. These anchor neighboring cells with one another for like physical strength. And then finally, gap junctions, which I think are a bit high yield, allow for communication between cells. And these we think of them when we think about cells that want to coordinate their activity. So the muscle cells in the heart, again, we'll talk about this in our biology playlist when we talk about heart physiology. So the muscle cells in the heart want to coordinate their activity, their contractions, so they're going to have these gap junctions between them so that they can transfer molecules and ions in between one another so that they can contract in a coordinated manner. And this is just the structure of gap junctions. I thought I have to include this because I actually did research as an undergrad, as a pre-med, related to gap junctions as part of a disease called Sherry-Copp-Marietou disorder. You don't need to know anything about this for the purposes of the MCAT. You'll learn all about it in med school. But essentially, you can see that the structure of a gap junction is composed of these 6 monomers. So each monomer is called a connexin. When they're all together, all 6 of them, you call them connexons. And these compose the gap junctions. All right, now let's talk about the concentration gradient. And the concentration gradient, although pretty straightforward, is very high yield on the MCAT. Essentially, molecules or solutes want to move down their concentration gradient, that is from high to low concentration. And that process would be a spontaneous process. It's a passive process, does not require energy. If you're thinking of in terms of thermodynamics, it has a negative change in G. Whereas the opposite of that process, if you wanted to transport molecules from areas of low to high concentrations, that would be an active process. So you would need to add energy for that process, such as in the form of ATP. And one important driving factor over here is entropy, and we'll talk more about this in our chemistry playlist, but essentially molecules, as they move down their concentration gradient, they are increasing their randomness. So you can think of this, if you put like, I don't know, a teaspoon of salt in a cup of water, it's not going to stay clumped up, ordered down in the bottom of the cup. It's going to want to spread out across the entire cup of water, and that is the increase in randomness or increase in entropy. Next, let's move on and talk about simple diffusion. So simple diffusion is what we've been discussing thus far. So the idea is you put a solute in a cup of water, it's going to spread all across. And the process of simple diffusion increases as you increase temperature. So if you increase temperature, the rate of simple diffusion will increase. Now, if you want to apply it to biological systems, it's across this membrane, and only molecules that are small or unpolar, like uncharged, can cross the plasma membrane via simple diffusion. So think about O2, CO2, steroid hormones. Although steroid hormones are not quite as small as O2 and CO2, because they're nonpolar, they can move freely across the plasma membrane in the process of simple diffusion. So no energy required over here. Next, let's talk about osmosis and tonicity. So essentially, osmosis is the idea of simple diffusion applied to water. So water is, like other solutes, is going to want to go down its concentration gradient, but I don't think that's the best way to think about it. Just think about it as water is going to go to the area with higher solute concentration. So in other words, the solute is going to suck water. So wherever you have more solute, that is going to suck the water. And these are examples over here. So you have a hypertonic solution. When you put like a cell in a hypertonic solution, what's going to happen is the outside solution has higher solute concentration. So it's going to want to suck the water out of the cell, and the cell is going to shrink. In an isotonic solution, it's not that you're not going to have any water movement. You're still going to have water flowing in and out of the cell. However, the net effect is zero. So the amount of water that flows into the cell will flow out. And that's because the concentrations are equal in solution and inside the cell. Now, moving on to the hypotonic solution. If you put a cell in a hypotonic solution, essentially the solutes inside the cell are higher concentration compared to the solution outside. So what's going to happen is the cell is going to want to suck water from the surrounding solution, and it's going to swell, and eventually it can actually burst if it sucks up too much water. Now, a good way to memorize this, because these are a bit tricky, is that hypotonic, so you have that O over there, hypotonic solutions want to make a big O of the cell, and that's essentially the idea of swelling up the cell. Osmotic pressure is another idea related to osmosis. So as you can see right here, if you get like a U-shaped tube and you put a semi-permeable membrane in the middle, and then you have water on either side of it. Now, what they're doing here is they're adding glucose to one side. Now, as we've discussed with osmosis, water is going to want to go towards the side with the higher solute concentration, so it's going to flow this way. Now, you might be asking yourself, well, why doesn't the glucose just flow across the membrane via simple diffusion? Well, that's because this is a semi-permeable membrane, so it's going to allow water but not solute to flow across it. But I did want to introduce this concept to discuss this equation. So pi is the osmotic pressure equals I mRT. R is the gas constant. M is the molar concentration. And by the way, you don't have to apply math to this equation, but you have to understand it conceptually for the MCAT. M is the molar concentration. So that makes sense. You increase the molar concentration, you're going to increase the osmotic pressure. T is temperature, same thing, that's pretty obvious. If you increase the temperature, as we mentioned earlier, you're going to increase diffusion and increase osmotic pressure. Now, I is what I really wanted to point out. I is the number of particles that your solute will dissociate into. So you might think that if you were to add like glucose to a solution versus table salt, that maybe glucose, because it's a heavier molecule compared to like NaCl, very small atoms, like glucose is probably going to have more of an osmotic pressure. However, it turns out that salt, table salt, will actually have a higher osmotic pressure. And that's because it doesn't rely on the details or the structure of the solute. What really matters is how many particles you're going to get out of that solute once you put it in solution. So glucose, you're only going to get one molecule or one particle, which is the glucose particle. However, when you put salt, you're going to get two particles, sodium and chloride. All right, now let's shift our attention to facilitated diffusion. Facilitated diffusion, we briefly introduced it when we talked about ion channels. It essentially needs proteins to allow for the transfer of ions or bigger molecules across the plasma membrane. So we have two different types of proteins that can be involved over here. You have channel proteins and carrier proteins. So you can see an example of a channel protein over here. It spans all the way through the plasma membrane, and it's essentially always open, so solutes can continue to flow down their electrochemical gradient in the case of ions, for example. Whereas if you're looking at the carrier proteins, they're more like revolving doors. So essentially at one end you have to have the solute bind to the protein, and then you have like a 3D conformational change, and then it'll turn around and release it on the other side, as you can see right here. So you can think of it as like a revolving door. You enter on one side, you flip the door, like you do that conformational change, and then you exit on the other side. All right, so now let's talk about active transport. So thus far, we've been talking about mechanisms that involve passive transport, where you don't need to add energy into the system because we were flowing from high concentration to low concentration, so down the concentration gradient. Well, in the case of active transport, you need to add energy because you're going against that gradient. So now we're going from low concentrations to high concentrations. It's like earlier we were rolling a ball downhill. You don't really need to add energy over there. However, when you're pushing the ball uphill, you need to add energy. And essentially we have two different types. We have primary active transport and secondary active transport. Primary active transport is where the cell uses energy, the energy of ATP, to transport an ion or a molecule against its concentration gradient. So one example is the NAK ATPase pump. We'll talk more about this in subsequent slides, but the idea is that you have higher concentrations of sodium out of the cell versus in the cell, but the cell wants to maintain that. So we constantly want to push sodium out of the cell, and to do that, we need to use ATP. Next, we have secondary active transport. This uses energy that has already been stored in a gradient. So for example, when you maintain that gradient of sodium with ATP, you're essentially storing energy over there. So if there's any molecule that you want to drive against its concentration gradient into the cell, you can use the energy stored in sodium to drive it in. One example of this is the Na, or the sodium glucose co-transporter. Essentially, the cell might have already a high concentration inside of it, but it's still hungry. So you still want to pump more glucose into it against glucose's concentration gradient. Well, what you're going to do is allow sodium to flow down its concentration gradient, so from high concentrations out of the cell to low concentrations into the cell, and that energy will allow you to drive glucose with sodium, but glucose is going against its concentration gradient. And the idea here is that this is a symporter because both solutes are going in the same direction, whereas if you are using one solute to drive another solute against its concentration gradient and they're flowing in opposite directions, then we call that an antiporter. All right, now let's talk about endocytosis and exocytosis. These are pretty straightforward concepts. You just need to memorize these terms. So endocytosis is where you engulf a bunch of particles or larger particles using a vesicle. So a common example is macrophages will engulf bacteria via the process of endocytosis, specifically phagocytosis, where we call it like cell eating, where you internalize large particles or even bacteria. Pinocytosis is where the vesicle will be filled with fluids, so it's like cell drinking. And then receptor-mediated endocytosis is to target a specific protein or a specific particle to drive it in. So you're more specialized over there. Now, exocytosis is essentially the reverse of endocytosis, where you allow particles from within the cell or fluids from within the cell to exit the cell via vesicles. One common example over here is the neuron terminal. So what you have is these neurotransmitters, and more on this in the biology playlist. The neurotransmitters are being released via exocytosis. So you have them in vesicles here, and they're released by fusing with the plasma membrane and then exiting into the synaptic cleft. All right, now let's talk about the last major topic here, which is membrane potential. So membrane potential is the electrical potential difference across the plasma membrane, so inside the cell relative to outside the cell, and it's maintained by an unequal ion distribution across the membrane. This is how the membrane potential is established. So this is our plasma membrane. This is the outside of the cell, and this part is the inside of the cell. You can see that you have potassium leak channels. These are transmembrane proteins across the plasma membrane. And then inside the cell, you can see a bunch of these red dots represent potassium. They're positively charged. And the blue, bigger circles are proteins which carry negative charges. And let's assume at the very beginning of this thought experiment, you have zero millivolt potential difference across the plasma membrane. So what's going to happen here is potassium naturally is going to want to leak through those potassium leak channels down its concentration gradient, which we also call a chemical gradient. So potassium starts to leak out, and you'll notice that as potassium starts to leak out, the potential difference, the membrane potential, will start to become more negative. Why is that? Because you're losing positive ions, positively charged ions, and you're maintaining the negatively charged proteins inside the cell. So it starts to become like, let's say, negative 10 millivolts. Well, what happens at that point is that you have two driving forces at play as it relates to potassium. Not only is it the chemical gradient that wants it to move outside, down the concentration gradient, but now you have an electrical gradient. Because this is now negative 10, there's going to be some tug of war going on where potassium wants to leave, but at the same time, the negatively charged inside of the cell wants to pull potassium back into the cell. But at this point, the concentration gradient, the chemical gradient, is still the winner. So it's still pulling out more potassium to leave the cell than the amount that's entering into the cell. Well, at some point, you'll eventually reach a membrane potential of negative 90, at which point you've established the electrochemical equilibrium, meaning that now the electrical gradient pulling potassium into the cell is equal in force to the chemical gradient pulling potassium out of the cell. So it's not to say that you don't have any movement of potassium in and out of the cell, but the net movement is zero. So the same amount of potassium that leaves the cell will re-enter the cell, and it'll continue to happen. Now, this would imply that the membrane potential in our body for our cells is negative 90 millivolts. Well, it turns out that's not the case, and that's because we need to take into account not only one ion, which is potassium, but we need to take into account sodium, chloride, and other ions that may play a role as well. And we further need to take into account the permeability of these ions. Of course, the higher the concentration gradient and the higher the permeability, the more of a role these ions will play in establishing the membrane potential. And it turns out that indeed, potassium is the main driver of the membrane potential. And that's because when you calculate the overall membrane potential, it turns out to be negative 70, which is very close to the negative 90 millivolts associated with potassium. Contrast that with sodium, for example, which has a membrane potential of about positive 60. So it's a lot different than the actual membrane potential. And that's how we know that potassium plays the major role in establishing that membrane potential. Now, the way we calculate it, you have this Nernst equation which takes into account the ions without the permeability of the ions. However, when you want to apply electrical potential to biological membranes, you want to take into account permeability and you want to take into account multiple ions. So that's where the Goldman-Hodgkin-Katz equation comes into play. You should be familiar with the way it's written out over here. Sometimes it'll show up in passages, but you don't need to memorize the details, and you certainly don't need to calculate anything using it. All right, this is just the structure of the NAKAT paste pump. So you can see that sodium is in higher concentrations out of the cell, potassium is in higher concentrations in the cell. And what this is essentially doing is pushing potassium against its concentration gradient into the cell and sodium against its concentration gradient out of the cell. And that process requires energy because it is an active process using ATP. All right, now these are knowledge check questions. So I'm going to push through, but if you have any clarifications, you can reach out to me and I'll help out with explaining these. All right. Okay, so a lot of high yield concepts, a lot of memorization. Be sure to take advantage of our Anki cards. And if you have any questions, reach out to me and you got this. Go and crush that MCAT. Don't gatekeep these videos. If you have a friend who's currently studying for the MCAT, be sure to share the playlist with them. It really helps the channel grow and I would really appreciate it.